Protein synthesis is the process in the cell that is responsible for the creation of proteins. In this video, we will be going over that process. First, there is DNA. DNA is the genetic code responsible for giving instructions to the cell. It is stored in the nucleus. It is a double helix. It has a phosphate, sugar, in this case deoxyribose, and a base in each section. These sections are nucleotides. Also, there are 4 types of bases: thymine in red, cytosine in yellow, adenine in blue, and guanine in green. Thymine bonds with adenine, and cytosine bonds with guanine.
The process of protein synthesis begins with transcription. First, a DNA helicase goes along the DNA and separates the double helix into 2 individual strands. One of the strands will be used as a template strand. Then, mRNA, also known as messenger RNA, is created with assistance from an enzyme called RNA polymerase. The polymerase uses the template strand to help create the mRNA needed. Additionally, as you can see, the sugar is a different color. Instead of deoxyribose in DNA, RNA has ribose sugar. Another thing to point out is that thymine, which has been replaced by uracil in the RNA, after it is created, the mRNA pairs with the template strand. After the DNA is replicated in the form of mRNA, the mRNA goes outside of the nucleus through a nuclear pore and is sent toward the ribosome. mRNA is vital since it is too dangerous for DNA to go outside of the nucleus.
When a codon of mRNA enters the ribosome, the process of translation begins. A codon is made up of 3 nucleotides that codes for an amino acid. This is a tRNA. As you can see, the tRNA is an exact anticodon of the mRNA. Additionally, the tRNA carries an amino acid. The first codon, with the bases AUG, is coded for a starting amino acid called methionine. After the tRNA pairs with its corresponding codon of mRNA, the amino acid is released. Then, the ribosome moves down the line of mRNA and the process is repeated.
The amino acids released by the tRNA form together to create a polypeptide chain. At the end of the mRNA, the codon, coded UGA, signals to the ribosome to cease any additions to the chain. A protein has been created. Now a fully formed protein, it will be sent into the rough endoplasmic reticulum. It will be picked up by a vesicle, transported to the Golgi body, then transported to its final destination, either inside the cell or outside the cell, through the cellular membrane.